The cell cycle is a control system that creates two accurate copies, or daughter cells, from each mother cell. For cells to replicate and stay more or less the same size, they have to double in mass before they divide. To keep the same genetic material, the DNA must be copied too. The cell cycle is divided into 4 phases, called G1, S, G2, and M. During G1, or GAP1 phase, cells accumulate biomass until they are almost double their original size. Cells then commit to start. Start involves the activation of a central regulatory protein called cyclin-dependent kinase, or CDK, in an irreversible positive feedback loop. Now cells enter S, or synthesis phase. CDK turns on thousands of protein machines that make new DNA and rapidly churn out a copy of the genome. Once S phase is done, cells may pause again in G2, or GAP2 phase, to create more biomass, or correct any errors that happened when copying the DNA. Once everything is ready, M phase, or mitosis, ensues. The chromosomes condense and are captured by the mitotic spindle, which is a machine that pulls them to opposite ends of the cell. Finally, the cell divides between the 2 duplicated genomes, creating 2 daughters that are very similar to their mother, but not always exactly, each with an identical copy of the genome and half of the cell's contents. A final note: there are surveillance systems or checkpoints in place in case something goes wrong. If DNA copying doesn't complete on time, or if DNA damage occurs, then the DNA damage checkpoint can delay the onset of metaphase. Furthermore, if the chromosomes are not correctly attached to the spindle, the process of chromosome division is also delayed. Completing the cell cycle correctly is crucial for our bodies to live and grow.